Good morning, everyone. Everyone, let's quickly find our seats because we have a lot in store for today. As we make our way to our seats, let's just be thinking about the unit that we just finished. All right, let's have a quick 30-second conversation about the unit about safety and microscopes that we just finished last class. Go for it. Microscope, safety. All right, let's put a cap on that conversation. Who will share one key fact from their conversation about our last unit? Go for it, Justine.
You should carry the microscope properly by the arm and the base.
Great. We'll take two more. That's a great start. Who will be next? Tracy.
To use extreme caution when handling slides because they're made of glass.
Absolutely. And lastly, go ahead.
Make sure no one trips over your cord.
Great. So all key safety aspects that we've learned last unit, and we'll use that to apply right here in today's unit. So today is a really big day because we're starting a brand new unit. And real quick, show of hands, who in here really enjoys puzzles? All right, a few of us. All right, for me, I really enjoy puzzles, but I'm not very good at them. So I need all of our help here today to complete a unique puzzle. At each of our tables, we'll have a set of images. And these images are categorized in a specific way. Our job is to figure out how these pictures relate to each other. All right, this is a competition, so amongst the tables. So let's see who can complete this the fastest and see how these photos are sequenced. All right, so everyone on the edge of our chairs, ready, set.
Wait, one.
Great question. All right, these images are face down. All right, face down. All right, everyone, what further questions do we have before we get started? We want to have a fair advantage.
Have one that's face up.
Yep.
It's supposed to be that way?
No.
Yep, so thanks, Ms. LaRose. This is a great question. So just to clarify, we have two sets of materials on our desk. We have one that's face up, one is face down. We are focusing on only the images that are face down. What further questions do we have?
Should I move over there because there's lots of people over here too?
Yeah, thanks, Justine. Are we ready? On the edge of our seats, and the time starts now. Go. All right, what are we noticing here? Okay.
Break these down to a very small scale. Very broad.
Yeah. All right. So you're on fire here. So let's run with that. Oh.
Done.
Oh. All right. Let's go ahead and finish. All right. How did y'all organize these?
Smallest to largest or largest to smallest.
Okay. So which side? Let's pick a side.
This would be smallest store.
All right. Are we done over here? Great. So it looks like all the groups are finished. So let's take a second to think about what just happened. So who will tell us how their group organized these photos? Shoot up a hand. Kathy.
From big scale to small.
Big scale to small?
Yeah. So
Who agrees with Kathy? Big to small. Okay, a few of us. All right. Why would it be big to small?
me still. Because you usually notice the bigger things first, and then you get in depth into it. So you don't notice like the smaller things first. You notice the big picture first.
Yeah, for sure. Let's flip down the coin. Who thinks that this is categorized smallest to largest? Shoot up a hand. All right. Why would we classify it small to large?
Well, this is my thought process on it, and to me, I started with the smallest because like cells are like the foundation to life, and so everything else just builds off of that.
Great. So this is the introduction of our brand new unit, which is plant biology. Each of these 9 different photos represents a different lesson. Alright, and so today we can see our first picture illuminated on the screen. Let's talk with our tables about a prediction about what this lesson might be about. Go for it. Good job. Alright, so I see most of us are finding the answers here. So who will take a stab at the prediction of what today's class is about? Heather.
The structures within plant cells.
Exactly. Who in here has ever had a lesson similar to this, like the organelles in a plant cell before? Okay, a few of us. Whether we are seasoned veterans and experts on plant cells, or if this is the first time we're hearing about this, at the end of this lesson, we'll all be on the same page. So imagine, like earlier, those puzzle pieces, right? Each of the puzzle pieces are coming together today in a plant cell, and by identifying these two objectives. So by the end of this lesson, our goal here as a class is to, with using a word bank, label 9 components of a plant cell with 100% accuracy and explain the function of each plant cell. So let's think about each component of a plant cell as a unique puzzle piece coming together to paint a full picture of a cell. Each has a very specific groove and function, and each can be identified in a very specific way. So for each organelle or plant characteristics, we'll focus on 2 items. We'll focus on how we identify it and how, what is the function. So once again, how can we identify it, and what is the function of this organelle? So we have 9 of those, 9 different components of a plant cell, and for each of the 9 components, we'll identify the function and how to identify it. So let's get started. All right. So this is an average plant cell. This is what most plant cells have in the organelles inside. Let's talk with a partner this time about some of the things that we see inside that we might already know the name of. Go for it. See, you don't have a partner, so I'll talk to you. So anything in there that you notice? Chloroplast. Okay, what does a chloroplast do? Alright, let's bring all of our conversations back in from our partners. We're going to throw them in a funnel them down to get started. So now, Mr. LaRose, we have that piece of paper that was up, and we can notice that it's the same image on the screen. So everyone, let's grab one of those sheets of paper. Go ahead, everyone have one? Cool. Let's go ahead and put our name, date, and period on these. This will signify as our note-taking page. This is our art canvas to capture any notes relevant. So be sure, as we go through, to identify how to identify the plant organelle and what is the function. So let's get started. By the way, just a secret, this is in alphabetical order. So let's start with this cell. If we see the arrow down here, what organelle are we pointing to? Anyone want to take a stab at it? Shout it out.
The cell wall.
The cell wall. So let's investigate the cell wall. Alright, so once again, in every organelle, we have the identification features and the function. So a cell wall is a thick, rigid, membranous brain of the cell. And the function is that it keeps the shape. So this cell wall is keeping the shape of this cell, and it's unique only to plant cells. So only plant cells have this really rigid structure that keeps the shape. Alright, so let's capture that down. This is also what allows plant cells to bond together. So in our first picture, we could see all the mini cells bonding together. That is due to the cell wall. Okay? Thumbs up if we're ready. Cool. All right, so guess number 2. Who thinks they know what this organelle is? Good. It's the chloroplast. All right, we have a lot of experts here. All right, so this is also a very unique organelle to the plant cell. So how we can identify it, this will be elongated or disc-shaped. And what's really, really cool about the chloroplast is that it contains chlorophyll. And chlorophyll is what allows photosynthesis to take place. Who will tell the class the 30,000 step overview of what photosynthesis is? Tracy.
So, photosynthesis is how a plant takes sunlight and uses it to help itself grow and then breathes out CO2.
Exactly. So without these chloroplasts right here, you and I, we wouldn't be able to breathe, because this is where photosynthesis happens. This is where all the energy from plants come from. All right here in the chlorophyll that's located in the chloroplast. So what questions do we have about chloroplast before we move on? All right, sweet. All right. So this is pointing to the jelly-like substance in between all the organelles. Anyone know what that one is? I think I heard it.
Is it
Jesse?
cytoplasm?
Cytoplasm, yeah, right on. All right, so cytoplasm. So this is, imagine, some Jello. It's that thick, membranous gel that's inside this plant cell, and it's basically where all these different organelles live. So that's the main function of a cytoplasm. It keeps all the organelles in location, and it gives it a place to move. Thumbs up? Cool. All right, here we go. So this is the big maze-looking item outside of the nucleus. This might be a little tougher.
It's the rough ER.
Yeah, it's the endoplasmic reticulum. So Heather mentioned rough ER. So the endoplasmic reticulum has 2 distinct parts. It has the rough ER and the smooth ER that are both located in this maze-looking tissue. The only difference is the rough ER has ribosomes and the smooth ER does not. And you might be asking, what the heck is a ribosome? Well, simply put, it's a protein that the plant cell uses. But the most important info that we need from the screen is that how to identify the ER, the endoplasmic reticulum. It's that maze-looking substance outside of the nucleus. And the function is that this helps package and sort different ribosomes and proteins to go in and out of the nucleus and also to the rest of the cell. Thumbs up? Awesome. All right, so this is the ER's BFF, his main squad, right next to him. What? You got it? Yo, y'all are killing it today. I figured this would be a lot harder, but y'all are so smart and so intelligent, so let's keep polishing that knowledge that we already have. So this is the Golgi body, also known as the Golgi apparatus or the Golgi complex. And it's like, I envision this as a stack of pancakes on one another. So it's very layered, and it's usually always next to the ER, the endoplasmic reticulum, because it, just like the ER, packages proteins, but its specific function is right here that we can know is that it's to export items from the cell. So the Golgi body focuses on taking any material that we have in the cell and exporting it out for the tissues, for the entire plant to use. Because the whole goal of a cell is to use what it needs and produce extra for the rest of the tissues and the muscles and for the body or the plant to work. All right, everyone should know this one. Let's bring it to the tip of our tongues. All right, and we'll say it on 3. 1, 2, 3. That's right. Let's say it with a little bit more excitement, people. You know this, all right? 3, 2, 1.
Nucleus.
That's right, and that's how exciting this organelle is. It is the essential organelle in the whole cell, right? So how we can identify it, it's the central part of the cell. It's what we can see under a microscope, and just foreshadowing what we're doing today, we'll be actually able to see these nucleuses in real life organelles using the information that we gained from our last unit with safety. We can use that to actually see this under a microscope. So who will read what the function of the nucleus is? Justine, thank you.
The nucleus controls many of the functions of the cell by controlling protein synthesis and contains DNA.
Okay. So DNA is what makes every living item unique. So every living item has a very different strand of DNA, and that is located inside the nucleus. But also the nucleus does a lot of other things, including protein synthesis, which is essential for a plant cell to make that protein to provide for the rest of the plant. All right, so now looking deeper inside the nucleus, anyone know what the middle organelle is? Well, let's get a drum roll for the nucleus. All right, so the nucleus is an organelle within the nucleus. So first we identified the nucleus, boom, check. Next, we can check in on the nucleus. All right, and this is very unique because this is where ribosomal RNA is produced. So think the big picture, the nucleus produces DNA, whereas the nucleus produces RNA. So let's pit stop here. Let's discuss with a partner one organelle that we have already talked about and their function. All right, we'll be ready to share 3 of the organelles that we've already covered from 3 different groups. So once again, an organelle, how to identify it and function. We'll be able to share 3 organelles from 3 different groups. What questions do we have? All right, let's pair up. Justine, what was your favorite organelle that we discussed so far? Okay. What does the cytoplasm do? Great. So let's take those 3 organelles. Remember, we are telling us how to identify it, the class, and also what the function is. Who will be first to share? Dana, thank you.
So we're gonna pick 3?
Just one. Good question. Just one. Yeah.
The cell wall.
Okay. And so tell us how to identify it and what the function is.
You look at the animal cell and you find the outside. So that's how you identify it, right?
Mm-hmm.
And then you, its function is like keep the cell together and let things move in and out.
Cool. All right. Thanks, Dana. All right. Who will be next to share? Kathy.
Alright, so we selected the chloroplast. We noticed also that there's 2 of them, so that's a good way to like be able to notice it because they look identical. And their function is, to help with photosynthesis.
Great. And who will be last to share? Heather. Tracy.
Wow.
So we discussed the cytoplasm and that it is jelly-like and it's all the extra space in the cell and it hosts the organelles of its function.
Great. All right. Any guess on this?
Are those the ribosomes you mentioned earlier?
Right on. So the ribosomes. So these are small organelles composed of RNA. All right. So quick question: where's RNA produced? That's right. And it has protein synthesis. Thumbs up. Cool. All right. This is the last organelle we'll be covering today before we get to our demonstration and our practicum. So this is very unique just to the plant cell. Any guesses?
Is it the water vacuole?
Yeah, right on, Dana. This is the water vacuole. So this is a really large membranous part of the cell that contains water. And so its essential function is to help maintain the shape of the cell along with supplying the cell with water. So today, we'll be able to do something a little bit differently. In the past, we were able to look at slides and microscopes under the microscope to use the safety skills we've learned. Now that we've learned about safety and microscopes today, we'll be able to prepare our very own slides. So up here, first I will demonstrate how to properly prepare a slide for our microscopes. So around the class, we have many different examples of plant cells. Up here, I brought 3. When we're preparing this slide, you're going to want to pick a sample that is very lush, that has some water to it, because the goal is to have a slide that you can see through. So it needs to be very thin, so you can see through it, so the light from the microscope can catch up these organelles like the nucleus and some other cells. So the materials we need is a scalpel, slides, and slide covers. So first, these scalpels, like we know already from our previous course, is that these are really sharp knives. So be sure to use all safety procedures when using these. There's a cover. Only remove the cover when we're using them. We have slides, and then we also have slide covers. So we will extract a sample from one of these specimens, put it on the slide, and cover it with the slide cover ready to put on the microscope. So I'll demonstrate that now. So remove our cover, right? I'm exposing the sharp knife, and then I'm just going to do a little scrape of the plant cell. So like I said, thinner the better. So we can see just a nice little sample. We'll place it on the plant cell, on the slide, and then we'll cover it. Okay? So this will allow for us, when we look at it under the microscope, we'll be able to see the plant cell and identify the key components of a plant cell. So let's get started. Here's the directions for what's coming up next. Boss. Thanks, Justine.
Yeah, no problem.
Thank you, Mr. Wasden.
How was my time?